A stark contradiction exists. Cancer cells aggressively divide to ensure survival, while Alzheimer's neurons initiate rapid, premature self-destruction. The QGM-LDDM framework applies a mathematical lens to these diseases, stripping away biological symptoms to measure hidden quantum states and electrochemical forces. Simulation data shows that both diseases exist in the exact same electrochemical pressure zone. This bar chart maps the force field data. A structurally degraded TP53 cancer protein exhibits a 46.9% thermodynamic repulsion. The highly conserved APP protein in Alzheimer's sits at 50.6%. The numerical gap between cellular immortality and rapid cellular decay is 3.6%. Despite producing opposite physical outcomes, their underlying thermodynamic profiles are virtually symmetrical. This symmetry indicates a shared quantum mechanism. Both diseases utilize the same physical tools, but execute different scripts. Because the energy states are nearly identical, the mechanism driving Alzheimer's cell death can be mathematically harvested and rerouted to attack solid tumors. This process involves isolating a specific neural frequency and inverting its phase to create a targeted therapeutic signal. Manipulating this shared thermodynamic baseline addresses the root physical state of the cell rather than the divergent symptoms of the disease. Mapping this baseline requires establishing the thermodynamic thresholds for survival and death using kappa-6 validated data. This plots force field thresholds. A healthy cell maintains 3.85% repulsion. Structural integrity collapses at 71.91%, the threshold for cell death. Cancer and Alzheimer's occupy the 46 to 50% middle ground, far from the baseline but below structural collapse. At this near-identical pressure, cellular fate is determined by a single variable, quantum fidelity. Fidelity measures the structural integrity of cellular hardware and its ability to process apoptosis signals. The cancer node registers a low fidelity score of 0.545, operating with structurally broken hardware. This degradation allows it to ignore the apoptosis signal and survive. The Alzheimer's node shows a high fidelity of 0.976, keeping its hardware pristine and highly conserved. This pristine hardware is hypersensitive to local environmental oxidation, which actively tricks the cell into initiating premature self-destruction. This creates two distinct therapeutic targets. We must pull Alzheimer's repulsion down below 3.85% and push cancer's repulsion over the 71.91% cliff. To push the tumor over that cliff, the engine extracts the destructive energy localized within the Alzheimer's model. The model isolates the 6.2 Hz theta band, which provides the neural resonance signature for Alzheimer's apoptosis. Within this band, the precise apoptosis signal amplitude is calculated at 0.083. Weaponizing this signal requires a mathematical phase inversion. Applying a pi shift rotates the frequency exactly 180 degrees along its axes. This transformation generates the inverted potential metric. The inverted Alzheimer's signal provides near-perfect alignment with the required signal needed to kill a cancer cell. There is a 0.99x match between the inverted 0.083 Alzheimer's signal and the 0.084 cancer target. This is not a chemical synthesis, it is the mathematical reprogramming of an existing pathological frequency. The inverted signal carries the repolarized kinetic energy of the Alzheimer's apoptotic process, now aimed at the tumor. The overactive apoptosis destroying Alzheimer's neurons becomes the blueprint for destroying oncology. The simulation tests this inverted signal against the G245S point mutation in TP53. The environment mimics an aggressive metastatic state with high oxidation and a low pH of 6.5. This mutation maintains a massive 99.8% BCL2 apoptosis resistance block. Breaking this resistance requires a therapeutic signal amplitude of 600.00. To reach this threshold, the signal must route through a precise structural vulnerability within the cell. Analysis of the top 10 therapeutic targets within the mutated structure reveals a consistent entry point. Guanine base pairs appear in every high-ranking therapeutic target. Guanine possesses the lowest oxidation potential of all DNA bases. This chemical property makes guanine an electrochemical weak point, susceptible to targeted structural disruption. The phase-inverted signal is routed directly into this guanine vulnerability. The targeted phase-inverted signal aggressively amplifies to a massive 960.80. In this simulation, the signal amplitude overcomes the 600 BCL2 resistance block, forcing even highly resistant metastatic cells into apoptosis. Scaling back from the DNA base pairs shows the macro architecture of the 18-qubit pipeline. This layer 7 LDDM formula calculates cellular fate by integrating force field variables and quantum states. As the signal hits the guanine target, variables recalculate in real time, tracking the cell's shift from survival to forced apoptosis. The process moves from sequence data to force field mapping, quantum extraction, and finally, phase inversion. The chaotic state of the disease snaps into a coherent crystalline alignment. Pathology here is a function of information coherence or decoherence within a quantum system. Computational biology exposes the hidden mathematical symmetries between unrelated diseases. A disease is a specific manipulation of cellular physical laws. Mapping these laws allows us to manipulate the state of the cell back toward health. The signals driving one pathology provide the mathematical blueprint to dismantle its biological opposite.